Hello and welcome to Agora. Agora is a platform for researchers to explore the results from data generated within the National Institute on Aging's Accelerating Medicine Partnership in Alzheimer's Disease, or AMP-AD consortia. These results include nascent Alzheimer's disease therapeutic targets that have been nominated by the AMP-AD teams, as well as results from consortia-wide analyses of RNA-seq data from postmortem brains. To explore the AMP-AD nominated targets, you can click on the View All Nominated Targets button. This takes you to a page with all 100 targets nominated by the AMP-AD consortia. You can see which teams nominated the target and how many independent nominations a given target has. You can click on the gene to see more information. In this case, I'm going to click on VGF, one of the most frequently nominated genes in AMP-AD. This will lead you to a summary page for this particular gene. At the top of the page is basic information about the gene, including its full name and a description of its known biological function. For genes that are nominated within AMP-AD, there is a button AMP-AD Nominated at the top of the screen that one can click upon to find out more information about why it was nominated. This takes us to a page with information about each of the teams that nominated the target, including a description of the team, why the target was selected, the predicted therapeutic direction, and the type of data and analyses used to identify this target. In this case, we have three teams: Mount Sinai School of Medicine, or MSSM, Emory University, and a team of investigators from the Broad Institute, Rush University, and Columbia University, or Broad Rush Columbia, that all nominated VGF. To see a full list of teams, we can go back to the landing page by clicking on the Agora logo at the top of the screen, and then clicking on the View All Contributing Teams button at the bottom of the screen. This page is a description of all 7 teams that have contributed to the analyses presented in Agora. So we see the Broad Rush Columbia team, the Duke team, a team from the Mayo Clinic in Jacksonville, University of Florida, and the Institute for Systems Biology, the Emory team, the Harvard and Massachusetts Institute of Technology team, the Sage Bionetworks team, and the Mount Sinai School of Medicine team. Now let's go back to the landing page and see what other sort of results we can explore. Similarly, I'll go to the top of the screen and click on the Agora icon to go back. Let's search for VGF again, but this time we're going to use the search bar. We see that VGF is one of the genes that's available. We also see it has a star next to it indicating it's a nominated target. Let's scroll down once the page loads to see what other results are available for VGF. We see below the description of the gene, there's a section called Summary of Evidence. If we click on the info box next to this, we'll see information about what each of these evidence fields indicates. So if I click on the info box, we see that AD genetic association is indicative of whether or not this gene has a significant genetic association based on the International Genomics of Alzheimer's Project genome-wide association study, whether or not it's a brain expression quantitative trait loci, and other fields accordingly. Some of these fields will be added in future versions of Agora. So for any of the ones that say coming soon, they will be populated in future releases. In the case of VGF, we can see that the AD genetic evidence is false, which means that there was no genetic association with VGF or the genomic regions near VGF identified in the IGAP genome-wide association study. However, for VGF, RNA expressions in AD brain is true. These results are available in Agora, and we'll see how to explore them next. Below the summary of evidence is a summary of the RNA-seq differential expression results from AMP-AD. These are results from harmonized analyses across 1,100 individuals and 2,200 brain samples. For reference, the first section shows the median expression in log counts per million in the 7 available brain regions. So this section right here. The info box indicates what the brain region abbreviations stand for. Anything above the red line is appreciable expression, and we can see that VGF is appreciably expressed in all the brain regions except the cerebellum. If I click on the info box, I'll see the definitions. DLPFC is dorsolateral prefrontal cortex, TCX is temporal cortex, and so on. Next is a differential expression result explorer. So right here. The box plot shows log2 fold changes for the selected gene in red in comparison to other genes. Note that the box plot shows results from one tissue and one statistical model at a time. By default, the base statistical model, AD diagnosis compared to controls, and the cerebellum are selected. If you hover over the red dot, you can see the summary statistics. We see that VGF is not differentially expressed in the cerebellum, but if we select a different brain region, such as temporal cortex, then it is differentially expressed. There are 7 tissues that can be selected, as well as 4 statistical models, including models that include sex by Alzheimer's disease diagnosis and age of death by Alzheimer's disease diagnosis interaction terms. If we change to the AD diagnosis by sex, in females only, we see that the result is still significant and in the same direction. To get an overview of the change in expression between controls and AD cases across the 7 available brain regions, there is a forest plot that we can inspect. This gives us an idea of how consistent the RNA-seq change is across brain regions between AD cases and controls. For VGF, we see a consistent downregulation of expression in AD brains compared to controls. In addition to the differential expression results, users can also explore genes that are similarly expressed based on co-expression analyses from the RNA-seq data. This is depicted with a network representation. So showed here. With the selected gene in red, VGF, and all the genes that are strongly co-expressed with VGF connected with edges. The more frequent an edge is connected across the 7 brain regions, the darker the edge. For example, ADCYAP1, which has darker edges, is co-expressed in more regions than EML6, which has lighter edges. Users can explore the neighborhood of the gene they are interested in to identify similar genes and see what functionality and other annotations these genes have. For example, I can select SYT12 and see that it is also a nominated target from the AMP-AD consortium, and I also will get a summary of the function of the gene. To go to the SYT12 page, I can click on any of the SYT12 links on the right. And now we're focused on the SYT12 gene, and all of the results that were shown before for VGF are now shown for this particular gene, including the description, the summary of evidence, differential expression, overall expression across tissues. You can see in this case it's expressed in cerebellum. The differential expression box plot, the forest plot also consistently down, as well as the similarly expressed genes network plot. If we go to the very bottom of this page, for this gene or any gene, we can get access to druggability data available for the 100 nominated nascent targets that has been provided by the AMP-AD industry partners. This can be accessed through Sage BioNetwork's data sharing portal, Synapse. So this is under the next steps section to learn more about the druggability of nominated nascent targets, please access a summary of their druggability here. But to access this data, you will need a Synapse account. To sign up for a Synapse account, you can click this link below. It will take you to a page with specific instructions on how to sign up for a Synapse account. There will also be a screencast going through each of these steps individually that is also available on our YouTube channel. Let's go back to the bottom of that page, and I'll just show you how to download this data once you have a Synapse account set up. So if you go to the bottom of the page, the druggability data for all of the 100 targets is available here. I click on this link. It takes me directly to the druggability data on Synapse. I'm not logged in. First I log in, and then I go to, I can download this file. There's a wiki that describes all of the fields in the file. So this file is just a CSV file with the gene names, the Hugo identifiers, the ensemble identifiers, and then a bunch of different druggability annotations. And the wiki has descriptions of exactly where those druggability annotations were derived from. So if I wanted to download, I would click the download button, and then it would be in my downloads folder on my computer. Let's go back to the portal. Finally, to learn more information about all of the analyses and Agora overall, you can go to the About page. If you have any questions, comments, or concerns, please do not hesitate to contact us by either clicking on the Contact Us link here on this page, or at the bottom of any of the pages, there's a Help button if you click on that will open up your mail client, and you can email us directly. Thank you.